Welcome to the deep dive. Today we're looking at something pretty exciting, the future of drug discovery.
It's moving so fast.
Right. And you're probably tuning in because you want to get a handle on the big shifts, the advancements shaping how new medicines get made, but without wading through super dense academic papers.
Yeah, we've done some of that legwork. We've sifted through quite a bit, especially looking at studies from organic process research and development, or OPR&D as it's often called.
Exactly. So our mission really is to pull out the key forward-looking ideas, the big paradigm shifts that are set to change how we find and develop treatments.
And what's really interesting is how different trends are sort of converging. We've got traditional pharma research, but now it's being seriously boosted by, well, digital tech and new ways of working in the lab.
It's not just small adjustments then.
No, not at all. I mean, it feels like a real sea change in how we approach the whole complex process of getting new drugs to people who need them.
Okay, so let's dive right in. AI, artificial intelligence, and machine learning. That comes up constantly. It sounds futuristic, but it's actually doing real work now, isn't it? I think I read about something called Eve.
Oh, absolutely. Eve AI is a great example. It shows how AI can find new uses for drugs we already have.
For repurposing them.
Exactly. Especially for things like neglected tropical diseases, which is huge because these are areas that often don't get a lot of funding.
Right, traditional research might not see the economic incentive there.
Precisely. So AI can potentially find treatments much more efficiently in these underfunded areas. The speed is, well, it could be transformative.
That's amazing, finding hidden value like that.
Yeah.
Now, pharma research, it just generates unbelievable amounts of data, right? Biology, pharmacology, clinical results. It must be overwhelming.
It really is, like finding a needle in a haystack, maybe several haystacks, and managing that, pulling out useful insights from all that diverse data, that's a major hurdle.
So AI helps with that data overload.
That's where it truly excels. AI and machine learning can handle these huge, messy data sets. They can integrate information even if it's sparse or comes from completely different experiments.
Okay.
And that allows for something called cross-dataset analysis. Basically, AI can spot subtle connections, patterns that a human researcher might just miss. This leads to, a much deeper understanding of the disease and how drugs might work.
So it connects dots we couldn't see before, potentially revealing new drug targets.
Exactly that. And it goes further. AI can actually help design new drug molecules, too.
From scratch.
Pretty much. Algorithms can look at vast libraries of potential molecules and predict which ones are likely to hit a specific target, designing candidates with very specific properties.
Wow.
But, and this is important, AI is a tool. It augments the researchers, it handles the heavy lifting with the data, finds patterns, but human creativity, that intuition, critical thinking, that's still absolutely central. It's a partnership.
That makes total sense. Human ingenuity directing the computational power. Okay, shifting gears a bit. What about the actual lab work, the hands-on chemistry? Is tech changing things there too?
Oh, definitely. Automation is massive. Think about automated synthesis and purification laboratories, ASPLs.
ASPLs, okay.
Yeah, these are basically robotic setups. They run chemical synthesis, purification, all guided remotely by researchers.
So a chemist in, say, London could run an experiment happening in a lab in San Diego.
That's the idea. It could dramatically boost efficiency, obviously, but also make global collaboration so much easier. You break down those geographical barriers.
It really does sound like the future, but also quite practical. Okay, moving along the pipeline. High-throughput screening, HTS. That's testing tons of compounds quickly, right?
That's it. You have these huge libraries of chemicals, and you screen them rapidly against a biological target to find initial hits, things that show some activity.
Yeah, that must generate.
Just vast amounts of data. Which brings us to chemoinformatics. You need computational tools to manage and make sense of all those HTS results.
Chemoinformatics.
Right.
How does that help specifically with HTS?
Well, after you get those initial hits, chemoinformatics tools help group them or cluster them based on their chemical structures, finding similar molecules. So instead of chasing down every single hit, which could be thousands, researchers can focus on maybe one or two representative compounds from each structural cluster. It's about working smarter.
That sounds much more manageable. Grouping similar structures lets you pick representatives. But I guess just looking at the structure doesn't always tell the whole story.
You've hit on a really critical point there. Relying only on structure can be misleading sometimes. You might lump together molecules that actually behave quite differently biologically, despite looking similar.
Right.
That's why understanding the structure-activity relationship SAR is so vital. How do small changes in structure affect what the molecule does? Chemoinformatics tools help analyze these SARs so you don't accidentally throw out a promising candidate just because it got grouped incorrectly based on a simplified structural view.
Got it. It's a more nuanced look than just shape sorting. Okay, let's talk manufacturing and quality. Obviously, quality control is paramount. How are digital tools helping there?
Automation is becoming huge here, too, for both quality and efficiency. Take Raman spectroscopy, for instance.
Raman.
It's being used more and more for real-time online measurement of, say, the drug content while it's being manufactured, like in extruded drug films.
So checking quality during the process, not just at the end.
Exactly. That's the core idea behind process analytical technology, or PAT. You build quality into the process by understanding and controlling it better. Digital tools are also allowing for much tighter control over basic steps like granulation, making powder stick together for tablets.
Oh, yes.
Better control there means more consistent, higher quality medicines in the end.
It's proactive quality control, baked right in. Makes sense. What about this continuous manufacturing idea? That sounds like a big shift from making drugs in batches.
It really is. Instead of making one batch, stopping, cleaning, then starting the next, continuous manufacturing uses integrated systems where everything flows, well, continuously.
And the benefit is?
Potentially much better process control. That usually translates to consistently high product quality and often improved safety, too. And, regulatory bodies like the FDA are actually encouraging companies to adopt these technologies.
Interesting. So we have these big trends: AI, automation, HTS, continuous manufacturing. But how does this stuff actually play out day to day? This is where those OPR&D studies you mentioned come in, right? Giving real examples.
Precisely. OPR&D is full of case studies showing these things in action. For example, you see lots of work on controlling crystallization. Things like seeding strategies, careful temperature ramps, choosing the right solvent, all to get the drug substance in a stable, bioavailable crystal form.
Controlling the crystal structure, the polymorphism. We talked about how crucial that is before.
Absolutely critical for efficacy and safety. And OPR&D papers detail how companies tackle this for specific drugs like fenofibrate, outlining exactly how they ensure they get the right crystal form every time.
Real-world problem solving.
Exactly. And you also see loads of examples of advanced chemistry being used at scale, like transition metal catalyzed couplings, powerful reactions for building complex molecules. OPR&D papers often show detailed reaction schemes, like scheme 10.1 or 10.2 you might find in some sources, illustrating how these are used in actual large-scale pharmaceutical synthesis.
So translating cutting-edge chemistry into something manufacturable. What else did these studies show?
Oh, lots on optimization, refining reaction conditions, making the workup procedures more efficient, like studies showing how to drastically reduce the amount of silica gel needed for purification. That saves money and is better for the environment.
Practical improvements.
Yes, and also novel synthetic methods, like new ways to do reductive aminations, which is a common reaction type. OPR&D publishes these new improved methods.
So it's not just finding new reactions, but making existing ones better, cleaner, cheaper. And you mentioned analysis, characterizing the molecules.
Definitely. Many OPR&D studies report incredibly detailed characterization of intermediates the molecules made along the way to the final drug, using techniques like NMR, FTIR.
To really understand the process.
Exactly. Understanding the reaction pathway deeply. There are examples where they meticulously characterize specific intermediates, like a certain carbamate or pyridinium salt, providing that crucial quality data. Plus, you find completely new synthetic routes or purification methods for drug candidates, like preparing a complex molecule called compound 8A.
It sounds incredibly detailed.
It is, and sometimes quite innovative too, like using oxidative dearomatization to build really complex structures that were hard to make before.
It's amazing to see that level of detail and innovation in process chemistry. Now, before manufacturing, there's preclinical development. Are these tech trends hitting that stage too?
Yes, they are. I mean, the bedrock is still rigorous preclinical toxicology studies for safety, of course. We've covered that before. But digital tools and better molecular understanding are making an impact.
How so?
Well, take CYP inhibition screening. This is done in vitro in the lab during lead optimization. It helps predict if a drug candidate might interfere with metabolic enzymes in the body, causing drug interactions later on.
So you can design out potential problems early.
Exactly. By understanding how a molecule might inhibit key enzymes like CYP3A4, which metabolizes many drugs, researchers can tweak the molecule or select a different one to minimize that risk way before it gets near a patient.
Proactive safety design. And what about how the body handles the drug? Pharmacokinetics.
Absolutely crucial early on. Things like absorption. How well does the drug actually get into the bloodstream? That's fundamental. Medicinal chemistry teams often face really tricky synthesis challenges at this stage, sometimes dealing with rejuicemers.
Molecules with the same parts, just arranged differently.
Right. And getting the right isomer with good pharmacokinetic properties can be a major hurdle. Overcoming those synthesis challenges is a big focus in preclinical work.
Okay, so it really feels like the whole landscape is shifting, from AI finding leads to robots in the lab, smart screening, digital manufacturing.
That sums it up pretty well. All these trends, often highlighted by the practical research in OPR&D, they're all aiming for the same thing, making drug discovery faster, more efficient.
And ultimately better medicines.
Ultimately, yes, getting better, safer, more effective medicines to patients who need them, that's the goal.
This has been really illuminating. Thinking about all these advancements, AI, automation, the detailed process chemistry from OPR&D, it really does make you wonder what's next. What new doors might open for personalized medicine or for tackling diseases we currently think of as, well, untreatable.
It's a lot to think about, definitely. The potential is huge, but so are the challenges and perhaps some ethical questions down the line.
Definitely food for thought. Thanks for joining me on this deep dive today.